Welcome to this lecture on the principles of immunology. Today, we are going to explore one of the most fascinating and critical systems in biology, your body's adaptive immune system. Specifically, we will dive deep into humoral and cell-mediated immunity. By the end of this session, you will understand exactly how your body identifies, fights, and remembers microscopic invaders to keep you healthy and alive. Let us start with humoral immunity. Imagine a medieval castle under attack by a swarm of invaders. To defend the castle without leaving the safety of the walls, the defenders use archers to shoot thousands of arrows into the battlefield. In your body, humoral immunity works exactly like this ranged defense system. Instead of archers, you have special white blood cells called B cells. And instead of arrows, they shoot tiny, highly targeted protein missiles called antibodies. These antibodies float through your body's fluids, historically called humors, seeking out invaders like bacteria and viruses before they can break into your vulnerable cells. Now that we understand the ranged defense analogy, let us look at the biological mechanism. Humoral immunity is absolutely critical for defending against extracellular pathogens, which are microbes that multiply outside your cells. When activated, B lymphocytes differentiate into plasma cells that produce and secrete specific antibodies. These antibodies bind to specific antigens on the surface of pathogens, marking them for destruction or neutralizing their ability to infect. Without this system, a simple bacterial infection could easily overwhelm your body because nothing would stop the bacteria from multiplying rapidly in your bloodstream. Before we look at the mathematics of antibodies, it is crucial to understand their long-term impact. The humoral immune response does not just fight the current infection, it also creates memory B cells. These cells provide long-lasting protection against future infections by the exact same pathogen. This is the exact mechanism that most vaccines rely on to protect you from future diseases. When you get a vaccine, your body is tricked into generating these specific antibodies and memory B cells. This ensures that if the real, dangerous virus ever enters your body, the defensive arrows are already waiting to neutralize it immediately. To understand how effectively these antibodies neutralize a threat, we must look at the chemical binding between the antibody and the antigen. This brings us to the dissociation constant, denoted as Kd. Conceptually, this formula tells us the ratio of unbound, free pieces to bound, neutralized pieces in the bloodstream. The numerator multiplies the concentration of free antigens by the concentration of free antibodies. The denominator is the concentration of the bound antigen-antibody complex. If the value of Kd decreases, it means the denominator is getting larger. Physically, this tells us the antibody binds very tightly and effectively to the antigen. Conversely, a high Kd means the antibody is weak and lets go easily. Now that we have our formula, let us see this with a concrete example. Suppose a patient has a bacterial infection. Laboratory tests show the free antigen concentration is 2 × 10⁻⁶ molar, and the free antibody is 3 × 10⁻⁶ molar. The bound complex is 6 × 10⁻⁵ molar. We need to calculate the dissociation constant to see if this antibody is strong. First, we substitute our given values into the numerator and denominator. Multiplying the free components in the numerator gives us 6 × 10⁻12. Dividing this by the bound complex concentration yields a final answer of 1 × 10⁻⁷ molar, or 100 nanomolar. Because this number is very small, it physically means the antibody binds tightly, providing a strong immune response. Having calculated the binding strength, let us visualize what this actually looks like in the body. In this diagram, you are looking at the humoral immune response in action. On the left, we have the large B lymphocyte. Notice the arrows indicating that it is actively secreting those Y-shaped molecules, which are the antibodies, into the light blue extracellular fluid. On the right, you can see rod-shaped bacterial pathogens. The key interaction is happening where the Y-shaped antibodies physically attach to the surface of the bacteria. This diagram perfectly illustrates how antibodies neutralize threats in the fluid before they can ever breach your cells. While humoral immunity is powerful, what happens if the invaders manage to sneak past the archers and hide inside the castle houses? The antibodies outside cannot see or shoot them anymore. This brings us to cell-mediated immunity. To clear out hidden invaders, you need specialized soldiers who go door to door, engaging in hand-to-hand combat to eliminate the compromised houses. Instead of using floating antibodies, this system uses specialized white blood cells called T cells. These T cells physically hunt down and destroy your own body cells that have been infected by viruses or have turned cancerous, ensuring that threats hiding inside your cells are completely eliminated. Now that we understand the close quarters combat analogy, let us examine the cellular mechanism. This system is absolutely vital for fighting off intracellular pathogens, such as viruses, which must hijack your own cells to replicate. Because antibodies cannot cross cell membranes, cell-mediated immunity is our only defense here. When a virus infects a host cell, the cell displays pieces of the pathogen on its surface. Cytotoxic T cells recognize these displayed pieces and bind directly to the infected cell. Once bound, the T cell releases toxic enzymes that trigger apoptosis, which is programmed cell death. This destroys the infected cell and the virus replicating inside it. Before we calculate how fast these T cells multiply, we must note two other critical roles of cell-mediated immunity. Because this system is designed to destroy abnormal host cells, it is the primary mechanism your body uses to detect and destroy cancer cells before they can form dangerous tumors. It constantly patrols for cells that look wrong. However, this same powerful system is also responsible for rejecting transplanted organs. When a patient receives a new kidney or heart, their T cells recognize the donated organ cells as foreign and attack them, which is why transplant patients must take immunosuppressant drugs. When a T cell recognizes an infected cell, it does not just attack and stop. It rapidly clones itself to build a massive army, a process called clonal expansion. This follows the mathematical rules of exponential growth. In this formula, N of T represents the total number of T cells present at a given time. N0 is the initial tiny number of T cells that first recognize the infection. The exponent divides the total time elapsed, T, by the doubling time, TD, which is how long it takes one cell to divide into two. Physically, if the doubling time decreases, the exponent grows faster, meaning the T cell army multiplies much more rapidly to overwhelm the viral infection. Let us apply this exponential growth formula to a real scenario. A patient is infected with a novel virus. Initially, only 50 specific T cells recognize the antigen. They divide with a doubling time of 12 hours. We need to find the total army size after 5 days. First, we must ensure our time units match. We convert 5 days into 120 hours. This is a crucial step, as mixing days and hours ruins the calculation. Next, we substitute our values into the equation. Dividing 120 by 12 tells us the cells undergo exactly 10 rounds of division. 2 raised to the power of 10 gives a massive multiplier of 1,024. Multiplying this by our initial 50 cells gives a final answer of 51,200 T cells. Physically, this shows how the immune system scales from almost nothing to a massive fighting force. Having calculated the size of the T cell army, let us look at how an individual soldier fights. In this diagram, you see the close quarters combat of cell-mediated immunity. On the left is the cytotoxic T cell, physically bound to the larger virus-infected host cell on the right. Notice the small particles being released between them? These are the toxic enzymes, perforins and granzymes. On the far right, you can see the result of this chemical attack. The host cell's membrane is fragmenting and breaking apart, which is labeled as apoptosis, or programmed cell death. This diagram perfectly captures how T cells sacrifice infected cells to stop viruses from replicating. We have now covered the full scope of this topic. Let us review the key takeaways. First, humoral immunity acts as your body's ranged defense, using B cells to shoot antibodies that neutralize extracellular threats in your fluids. Second, cell-mediated immunity acts as your close quarters combat team, using T cells to hunt down and destroy intracellular threats hiding inside your own cells. Finally, remember that these two systems do not work in isolation. They are a perfect complementary team. By combining ranged and close quarters defenses, your adaptive immune system ensures that pathogens have nowhere to hide, providing you with comprehensive and total protection against disease. Excellent work today!